So, as we just discussed, potential cures could command significant market share and R&D investment because of the potential promise that they could provide. So, we just discussed that based on ARC's research, gene editing and gene therapy, companies could grow 54% at a compound annual growth rate, and that could scale from 130 billion to 1.1 trillion. So, let's unpack some of those innovations that will contribute to those numbers with Professor David Liu. So I'm going to do a short introduction because we could do a much longer one. But David is a chemist, a biologist, and a core faculty member of the Broad Institute, as well as a professor of chemistry, chemical biology at Harvard University. He's an investigator of the Howard Higher Medical Institute, and David's lab has been instrumental in gene editing and in the gene editing world by developing base editing and prime editing, which is what we just discussed, also precise mitochondrial editing and other technologies. He founded or co-founded several companies, including Editas Medicine, Beam Therapeutics, Pairwise, Plants, Prime Medicine, Chroma Medicine, and Exotherapeutics. So thanks for coming on, David, and looking forward to the conversation.
Yeah, likewise. Thanks for having me.
Great. So let's just jump right into it. So CRISPR obviously has contributed to this explosion of gene editing by creating just this more efficient way of editing cells. As an academic, you're using CRISPR, but you're also using other nucleases in the lab, like zinc finger nucleases and TALENs. Can you tell us a little bit about why CRISPR, and I don't want to leave the listeners, so why CRISPR may be the easiest to use, or tell me that it's not?
Yeah, I mean, I think there's a broad consensus among the practitioners that if you want to engage a specific DNA sequence, CRISPR is the easiest way to do that. And of course, if you want to edit a DNA sequence, you have to start by engaging that sequence. Proteins are commonly 3D blobs that don't normally recognize individual DNA bases in a programmable digital way, but instead they do so by shape fitting for most DNA recognizing proteins in nature. And what that means is that there isn't a simple DNA recognition code whereby you can change one protein letter and recognize one DNA letter. So the oldest class of gene editing agents are arguably the meganucleases or hominidonucleases. These are proteins that cut the DNA double helix, but they each evolved in nature a specific shape to cut a specific DNA sequence. And since gene editing applications require that we cut DNA sequences of our choosing, not nature's choosing, if I give you a list of, say, 100 sequences that I want you to cut to treat some genetic diseases, for example, it would likely take my lab decades to generate 100 hominidonucleases that could cut those 100 sequences. So zinc finger nucleases were the first programmable DNA cutting enzymes. Being programmable, you can determine the sequence of a zinc finger nuclease that will cut a specific DNA site of interest. But the challenge is that the code is not quite as simple as a lookup table as you might want, and it still takes some amount of optimization to generate a zinc finger nuclease that cuts really efficiently and selectively a given DNA sequence. So if you gave my lab a list of 100 DNA sequences and asked us to make 100 zinc finger nucleases to cut those 100 DNA sequences of your choosing, it would probably take my lab years to do that, although I think it would be successful. The TALENs are also programmable DNA binding proteins, but here the beauty is that the code is very simple. In fact, it's just 2 protein letters in each tail repeat that determine a single DNA base that's recognized. So if you were to give me that list of 100 sequences and ask me to make 100 TALENs that cut those 100 sequences, it would be expensive. It wouldn't be easy to make. It would be somewhat labor intensive to construct, but I think our lab could probably do it in months as opposed to years. And then finally, to put things in perspective, CRISPR-Cas9 was such a breakthrough because the code to recognize a DNA sequence is purely determined by a guide RNA. And guide RNAs pair with DNA following simple Watson-Crick rules, where A binds to T and G binds to C. So if you gave me well, actually, I have a better example because we've published examples. We performed 38,000 CRISPR edits using a library of guide RNAs and different libraries of base editors, uh, and making those 38,000 base editors, CRISPR agents, to edit each of those 38,000 sites took less than a year of effort. It really has been not a small change, but really a transformative acceleration of our ability to programmably access DNA sites of our choosing, which ultimately is critical for many of the applications of gene editing, especially in therapeutics.
And what's pretty incredible is that not only did you take that advance, you took CRISPR and you expanded what was possible with CRISPR in your lab. So much of what we just talked about in the last presentation was really things that were developed in your lab. So base and prime editing, there are two discoveries that were found in your lab with postdocs, Alexis Comore and Nicole Gadelli, and also Andrew Anzalon. So can you discuss maybe the aha moment for these technologies, their importance, but also I've heard before that, it took Andrew Anzalon a very short amount of time to get from conception and idea to actually getting this to work. And so maybe that can also sort of show and illustrate what we're talking about,
Sure.
when we talk about this. And also just to set the scene, we just talked about a little bit the size, the PAM dependence, the lack of DNA double-stranded breaks, clinical variants, more of them being used. So feel free to expand on those or others.
Yeah. Yeah, so maybe it's important to take a step back and just to define some terms here. Gene editing is a general term that encompasses lots of ways that people make changes to DNA. It includes gene disruption, messing up genes. It includes gene correction, which is precisely fixing a mutated gene, like one that causes a disease, back into the normal healthy sequence. It includes gene insertion, which can be useful for gene therapy. So nucleases, as you mentioned, cut DNA. They're like molecular scissors that literally take a chromosome and break it into two pieces. Cutting a DNA with a nuclease is best for gene disruption because when you cut DNA, especially in mammalian cells, you usually generate a mixture of messed up gene products. That's actually exactly what CRISPR-Cas9 evolved to do in nature, after all, which was to cut viral genomes as a way of messing up virus genes to stop viruses from infecting bacterial cells. So disrupting genes can be messy, but it can be very useful for many research applications and even for some therapeutic applications, as demonstrated by some of the very encouraging clinical trial results that you mentioned that have come out over the past several years. But for most of the 6,800 or so, 6,800 or so genetic diseases that we understand are caused by specific mutations in DNA, we don't think we can really address most of those diseases by messing up the gene. The gene is already mutated that causes the disease, and most of the time, to best treat the disease, we think we need to precisely fix the mutation, to turn it back into the normal sequence, or maybe to turn it into a sequence that would be harmless or is benign compared to the mutated disease-causing sequence. And so that's really what inspired us to develop base editing and prime editing, that using a nuclease to cut DNA to try to serve as a starting point for precise gene correction is possible in certain kinds of cells in a dish, but turns out to be really difficult in therapeutically relevant cells. That's the process called homology-directed repair. So Alexis Comore was an incoming postdoc at the time, and she was searching for a project to start. And we iterated emails. This was in 2013. And I encouraged her to think about pairing a CRISPR-Cas9 protein to home in on a target sequence of DNA, but to use a disabled form of the CRISPR protein that no longer cuts the DNA, but is still programmable with a guide RNA. And instead of cutting the DNA, to integrate a deaminase enzyme that performs chemistry on an individual DNA base, converting that base to another base. And so 3 years later, she successfully developed the first base editor system, which directly converts C's to T's or G's to A's at targeted sites of our choosing, using CRISPR for its wonderful targeting mechanism and ease of targeting. And that was a real change in the way that at least our lab thought about how we could use gene editing to not just disrupt genes, but to actually fix them. Then a year later, or 1.5 years later, Nicole Godelli, that you mentioned, a former postdoc in the lab, developed the adenine base editor. That's actually the base editor that recently, a form of which recently received FDA clearance to begin a clinical trial led by Beam Therapeutics. The adenine base editor is a different type of base editor that performs another chemical reaction on different DNA bases, converting A's into G's or T's into C's. And then, as you mentioned, Andrew Anzalone, a third former postdoc in the lab, really brought a wonderful idea to the lab of not just using CRISPR to locate a DNA sequence and then perform an individual base chemistry on one base pair, but rather to directly write new DNA sequences into a target site in the human genome in a way that allows you to replace dozens of DNA base pairs with just about any sequence that you want to choose. So you can now make all the 12 possible base-to-base substitutions, as well as small insertions, small deletions, or combinations thereof. And that's why, as your analysis suggested, prime editing, which is the method that came out of Andrew's work in the lab, can, in principle, correct such a large fraction of mutations that are associated with those 6,800 genetic diseases. And as you pointed out, it actually only took Andrew 1 complete year. It was 1 year and 8 days, I think, from the time he set foot in the lab to the time we submitted the prime editing paper. Now, I think there are two major factors behind the fact that the development of prime editing was so fast. The most important of which is Andrew is a very, very special scientist, is just a world-class thinker and a world-class executor, and works beautifully with others. In fact, he enables all the scientists around him, helps them, and they help him. So it was really a perfect storm of Andrew's capability, his dedication, and the soundness of the idea that led to the development of prime editing. But I think the second contribution is really the one that you're trying to make and that your slides made beautifully, which is that the whole way science works, especially in academia, is that we build on the accomplishments and the discoveries of others. And the seminal work of Emmanuel Charpentier, Jennifer Doudna, George Church, Fun Zhan, and many others to decipher the mechanism and the logic and the capabilities of CRISPR is what allowed the development of prime editing to take place so quickly.
Yeah, it's pretty remarkable what he did in a year when you think about what others have accomplished in years. But so we're going to need to wrap up in just a minute. So unfortunately, last quick fire question. We talked a lot about our sort of research and the idea that by 2026 we should see the share of total R&D spending devoted to gene editing and gene therapy is growing from 3% to 17%. Based on your experience, what are you most excited about in the next 5 to 10 years in the field, given that there could be an expansion of R&D spending and therefore opportunity in the field?
So my answer has evolved over the past several years to be maybe more ambitious, more optimistic, encouraged by the data, by the fact that, as you've shown, there's been an exponential explosion of publications, of real advances, of clinical applications of gene editing technologies. So I think within 5 years, we will see a very broad and deep and diverse set of clinical outcomes that really validate the use of gene editing technologies to treat genetic diseases by actually correcting their root cause, both ex vivo and in vivo, in a variety of organs. I think that will be a landmark achievement in the life sciences, really a landmark achievement in the history of man, if you appreciate that our genomes have dictated so much of the aspects of our lives, including in many cases genetic destinies that limit our lifespan or the quality of our lives. And for the first time, we are able to correct the misspellings in our DNA that otherwise would have fated us to a far uh, uh, poorer existence with much more suffering for patients and their families. And then within 10 years, I'd like to think that gene editing in the clinic will hopefully reach a state of being both efficacious and safe enough that we will begin to use gene editing on a far broader scale. A great example of this is the collaboration between Verve Therapeutics and Beam Therapeutics to use base editing to treat hypercholesteremia by editing PCSK9. Initially, they, I think, very thoughtfully are choosing patients with an extremely high risk of coronary heart disease because of mutations that they have in the genes that regulate their cholesterol levels. But if that treatment is proven to be safe and efficacious, you could imagine expanding it to high risk people that maybe don't have the mutations that give them incredibly high cholesterol, but maybe they just suffered a heart attack. Or maybe they have other risk factors that make them at higher risk than normal for heart disease. And then if that proves to be safe and efficacious, then you get to this exciting possibility of wondering, well, if the leading cause of death in humans is heart attacks, is heart disease, and if all of us have PCSK9 genes, or almost all of us have PCSK9 genes that we can edit to reduce that risk, then is it worth thinking about putting a real dent in heart disease by editing a much broader fraction of the population? And that's really an exciting question, filled with all sorts of important ethical and societal implications that we'll have to think through very thoughtfully. But that could mark the beginning of the end of what is currently the world's leading cause of death. So that's just one example of many. You can make a very similar set of arguments for how an expanded application of gene editing could also be used to treat a wide variety of diseases, including Alzheimer's disease, other neurodegenerative conditions, certain forms of cancer, et cetera. So that's why this is a time unlike any other that I've experienced in my 23 years as a professor in the life sciences, one where this perfect storm of technologies, of molecular understanding, of AI, of DNA sequence information, and of gene editing technologies like the ones you've summarized have come together to enable us to finally take control of our genetic destinies.
Yeah, so you've just nicely outlined how gene editing has a really wonderful and bright future. So thank you for that. And just want to thank David so much for joining us today. It's always great to hear more about progress in the lab, and look forward to next time. Thanks very much.
Thank you for having me.